Welcome to G Fun Facts Online. In 1953, the world was introduced to an image that would forever change our understanding of life: the elegant, spiraling ladder of the DNA double helix. It's a symbol of scientific genius, but the story behind it is far more complex than a simple eureka moment. It's a tangled tale of fierce ambition, clashing personalities, and a monumental injustice that has taken decades to correct. This is the story of how the secret of life was unveiled, and how one scientist's crucial contribution was almost lost to history. By the middle of the 20th century, scientists were in a frantic race to answer one of biology's biggest questions: what molecule carries the blueprint of life? For a long time, the smart money was on complex proteins, but mounting evidence pointed towards a different, more mysterious molecule: deoxyribonucleic acid, or DNA. The challenge wasn't just knowing DNA was important, it was deciphering its three-dimensional structure. Whoever could do that would unlock the very mechanism of heredity. This scientific Grand Prix had contenders across the globe. In the United States, the legendary chemist Linus Pauling, a master of model building who had already cracked the structure of proteins, was closing in. His reputation alone created immense pressure on other research groups. The main competition, however, was brewing in the United Kingdom, split between two rival teams. At King's College London, a team led by Maurice Wilkins was using a technique called X-ray diffraction. This involves shooting X-rays at a substance and analyzing the scatter pattern to figure out its molecular shape. Wilkins had already produced some of the first decent X-ray images of DNA. In 1951, he presented these images at a conference in Naples, Italy. In the audience was a young, ambitious American biologist named James Watson. Seeing Wilkins' images ignited a fire in him. He was instantly obsessed with solving the DNA puzzle. Watson soon found himself at the Cavendish Laboratory in Cambridge, where he joined forces with Francis Crick, a physicist with a brilliant theoretical mind. Unlike the experimental, hands-on work at King's College, Watson and Crick's approach was to build physical models, like a complex Lego set, trying to fit the pieces together based on the available data from other labs. Their first major attempt was a disaster. Based on some information from a lecture, they hastily built a model with 3 intertwined helices. It was completely wrong, and so embarrassing that their lab director ordered them to stop all work on DNA. This brings us to the central figure whose role was misunderstood for so long: Rosalind Franklin. In 1951, Franklin, a supremely talented X-ray crystallographer with a PhD from Cambridge, joined Wilkins' lab at King's College. From day one, their relationship was poisoned by a simple misunderstanding. Wilkins was away when she arrived and assumed she was hired to be his assistant. Franklin, however, had been told she would be leading the DNA project independently. This immediate friction and lack of clarity created a tense and difficult working environment. Despite the frosty atmosphere, Franklin's work was meticulous and groundbreaking. She was a brilliant experimentalist. She discovered that by carefully controlling the humidity around the DNA samples, she could produce 2 distinct forms: a drier A form and a wetter B form. It was her work on the B form that would change everything. In May 1952, Franklin and her PhD student, Raymond Gosling, took an X-ray diffraction image of this B form DNA. The result was a picture of stunning, unprecedented clarity. It was labeled simply Photograph 51. The image was a perfect, unambiguous snapshot of a helix. The distinct X shape formed by the scattered X-rays in the center of the image was the undeniable signature of a helical structure. From this single image, a trained eye could deduce the angle of the helix and the distance between the repeating units. It was the key. However, Franklin was a cautious and rigorous scientist. She believed the path to absolute certainty lay in analyzing the more complex and detailed patterns from the A form of DNA. So she set Photograph 51 aside for the time being, choosing to focus her immediate efforts elsewhere. It was a fateful decision. The race for the structure kicked into high gear in January 1953, and this is where the story takes its most controversial turn. Without Rosalind Franklin's knowledge or permission, her colleague Maurice Wilkins showed photograph 51 to James Watson. For Watson, seeing that crisp X pattern was the light bulb moment. He later wrote, "The instant I saw the picture, my mouth fell open and my pulse began to race." All his and Crick's theoretical work suddenly had a concrete foundation. The helical structure was no longer a guess, it was a fact. The data pipeline didn't stop there. Watson and Crick also gained access to a confidential report Franklin had written for a Medical Research Council committee. This report contained her precise calculations and measurements derived from her X-ray work, including crucial details about the size and symmetry of the DNA molecule. Armed with Franklin's priceless, uncredited data, Watson and Crick raced back to their model building. They now had the final pieces of the puzzle. They combined the structural clues from photograph 51 with another key finding by the biochemist Erwin Chargaff. Chargaff had discovered that in any DNA sample, the amount of the base adenine, or A, always equals the amount of thymine, T, and the amount of guanine, G, always equals cytosine. Crick's expertise in diffraction theory and Watson's biological intuition came together. They realized the structure wasn't 3 helices, but 2. They figured out that the 2 strands ran in opposite directions, creating a double helix. The sugar phosphate components formed the outer backbones, like the sides of a twisting ladder. The crucial insight was how the rungs of that ladder were formed. The bases were paired on the inside, A always with T and C always with G. This specific pairing, known as base pairing, was the secret. It not only fit the width constraints from Franklin's data perfectly, but it also immediately explained Chargaff's rules. More importantly, it suggested how DNA could replicate itself. If you could unzip the 2 strands, each one could serve as a template for creating a new, identical double helix. In their landmark 1953 paper, published in the journal Nature, they famously understated the significance of this, writing that, "It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material." In that very same issue of Nature, papers from Wilkins' team and from Franklin and Gosling were also published. They served as the experimental evidence supporting the Watson-Crick model. But while their work appeared alongside the main discovery, all the glory, fame, and credit for the breakthrough went to Watson and Crick. In 1962, the ultimate scientific prize was awarded. James Watson, Francis Crick, and Maurice Wilkins jointly received the Nobel Prize in Physiology or Medicine for their discovery. Rosalind Franklin's name was not mentioned. Tragically, she had died 4 years earlier, in 1958, from ovarian cancer at the age of just 37. Her cancer was likely caused by extensive exposure to X-ray radiation during her research. The Nobel Prize is not awarded posthumously, a rule that has been heavily debated in her case. Even if she had been alive, many question whether the prize committee in that era would have recognized a woman's contribution on par with her male colleagues, especially given the way her role had been minimized. The official story of the discovery was largely shaped, and arguably distorted, by James Watson himself. In 1968, he published a personal memoir called The Double Helix. The book was a bestseller, praised for its candid, almost novel-like depiction of the scientific process. But it was also heavily criticized for its deeply flawed portrayal of Rosalind Franklin. Watson painted her as a difficult, uncooperative, and frumpy figure he condescendingly called Rosie. He characterized her as a mere assistant who hoarded her data and failed to understand its significance. This narrative cemented a public perception of Franklin that was entirely inaccurate, painting her as a technician rather than the brilliant scientist she was. The book was so inflammatory that both Francis Crick and Maurice Wilkins were furious with Watson's account and tried to stop its publication. In a later edition, Watson did add an epilogue where he admitted his initial impressions of Franklin were wrong and acknowledged the superb quality of her research. In the decades since, the tide has turned dramatically. Historians and biographers have pieced together the real story, revealing the sexism Franklin faced in the male-dominated scientific circles of the 1950s. She is no longer seen as a tragic, wronged heroine, but as an equal and independent scientific mind, whose experimental work was absolutely fundamental to the discovery. Her meticulous, high-quality data provided the essential evidence that allowed Watson and Crick to build their correct model. The question of whether her data was stolen is still debated. Some argue that in the competitive environment of the time, sharing information wasn't uncommon. But there is no debate that her work was used without her knowledge, and she was not given the proper credit at the time of the discovery. Today, Rosalind Franklin is celebrated as a scientific icon. Buildings, universities, and research grants now bear her name. Her story has been told in popular plays, most famously Photograph 51, bringing her life and work to a global audience. It's also important to remember that her scientific career didn't end with DNA. After leaving King's College, she went on to do pioneering research on the molecular structures of viruses, including polio. She laid the foundations for the entire field of structural virology. In a poignant epilogue, her colleague Aaron Klug, who continued her virus research after her death, was awarded the Nobel Prize in 1982 for the work she had started. The double helix is a beautiful, powerful symbol of life. But its history is a messy, human story of ambition, rivalry, genius, and injustice. It's a powerful reminder that scientific breakthroughs are rarely the work of isolated heroes, but the culmination of many people's efforts, not all of whom get to stand in the spotlight. The tangled steps to glory for the double helix serve as a permanent lesson in the importance of ethics, collaboration, and recognizing the contributions of all who paved the way to discovery.